Yep.
Yeah, okay, so then
You.
I'll begin now. Hello, everyone. My name is Snigtha Mohanraj, and I'm a sophomore at the Engineering and Science University Magnet School. And my project is called Implementing Non-Toxic Modified Biochar Enhanced Filtration for the Efficient Removal of Emerging Contaminants in Aqueous Solution. So firstly, I'd like to say thank you to Miss Alyssa Anderson. She was my school mentor, and she gave supervision and help with buying materials, and she guided me in using the school labs. And I had some outside lab work for the liquid chromatography mass spectrometry analysis, so I wanted to say thank you to Miss Sydney Schuster and Dr. Fabian Menges, who helped me in the search for the equipment and using the instrument itself at Yale University so that I could gather my data. And then Miss Kristen Arnini, who's the New Haven Science Fair mentor coordinator, who put me into contact with Miss Sydney Schuster and Dr. Fabian Menges, and of course my parents for supporting me. And so basically what this project was aiming to do was to determine an inexpensive, environmentally friendly, sustainable, and efficient method for removing harmful emerging contaminants from our water by utilizing doped biochar enhanced with metal oxide nanoparticles. I hypothesized that the most effective method would be achieved through using specifically coconut shell biochar doped with iron oxide nanoparticles. So, water contamination is a problem that I've been interested in for a long time because it's one of the most pressing issues. It affects everyone daily, no matter what, and it's found in almost all water sources. And so in 2019, I first targeted just microplastics in water and investigated natural coagulants. And then in 2020, I investigated metal oxides with an electromagnetic filter, again for removing microplastics from water. In 2021, I expanded this and targeted both microplastics and oil, and I designed the ferrosponge, which is a sponge coated in iron oxide nanoparticles and bentonite to remove both microplastics and oil. And through using the homemade spectrometer that I built, I was able to confirm that iron oxide nanoparticles are indeed very effective for contaminant removal, and this motivated me to utilize them in this current research as well. And so my current research investigates different biochars enhanced with metal oxide nanoparticles to remove emerging contaminants from our water. I tested different biomass materials, which were rice husk and coconut shells, to see how that affected the results. And then I tested two different dopants. They were iron oxide nanoparticles and manganese dioxide nanoparticles, since nanoparticles can increase the sequestration of contaminants by adding or improving properties of the biochar. And so then I tested the removal of different pharmaceuticals, pesticides, microplastics, and oil, because these are some of the most common and detrimental contaminants that we can find in a lot of water sources. So, what are emerging contaminants? So, this is an umbrella term that refers to water contaminants whose presence in our water has only been recently confirmed and investigated, but they've long been suspected to be in our water. Many water sources contain these emerging contaminants, and some examples are again are pharmaceuticals, pesticides, microplastics, and oil, which is why I targeted them in this project. They can be very toxic to aquatic organisms and the environment and can lead to a variety of detrimental health effects in humans. However, current water treatment processes are unable to remove them from our water because we have such limited knowledge on them. So my solution is to use biochar, which is a material produced by the pyrolysis of biomass with limited or no oxygen present. This material is mostly used agriculturally, specifically in soil amendment processes, because it can absorb and sequester pollutants and other chemicals in the soil. These sequestration properties are exactly why I wanted to utilize them in this project as well, since those properties were expected to transfer over for water contamination. I can modify this biochar by adding dopants, which can demonstrate a significant increase in contaminant removal because of the enhancement of the biochar's properties. So my project investigates coconut shell and rice husk biochars to see how the material affects it, and then two different dopants, which are iron oxide and manganese dioxide nanoparticles. The iron oxide has a high surface to volume ratio, meaning that there's more area available to attract and sequester the pollutants, and then there's a favorable structure for doping. And then the manganese dioxide is an effective adsorbent and has a strong stability. So six kinds of biochar were produced. This was undoped coconut shell and rice husk biochars, iron oxide nanoparticle doped coconut shell and rice husk biochars, and then manganese dioxide nanoparticle doped coconut shell and rice husk biochars. And in some of the graphs, you'll see that activated carbon was used as a control just to see a comparison as to how effective these methods are in accordance to the activated carbon, or AC for short. So these pictures show the iron oxide nanoparticle synthesis for the doped biochar, and these pictures show the manganese dioxide nanoparticle, again for the doped biochar. And then, so the contaminated samples, I tested 6 different contaminants. The first 2 were acetaminophen and ibuprofen, which are pharmaceuticals. The next 2 were DEET and glyphosate, which are pesticides. And then PET microplastics, since PETE is the most commonly used form of plastic, and then gasoline oil. So these pictures just show some of the contaminated sample preparation. And then each experimental biochar treated each type of contaminated sample. The biochar was added to the centrifuge vial containing the contaminated sample. It was centrifuged, the samples were filtered, and then the supernatant was taken to the corresponding analysis. So the first analysis was the liquid chromatography mass spectrometry, which is the outside lab work at Yale Labs. And using the lab software, contamination level of pharmaceutical and pesticide samples was recorded. The second analysis was microscopy, which microplastic contaminated samples were placed under a digital Wi-Fi light microscope connected to a software program, which is shown on the right, and the number of microplastic pieces in each sample was recorded. And then the third and final analysis was the light spectrometry, in which oil samples were placed inside the spectrometer and the relative light absorbance of each sample was recorded. It is important to note that absorbance isn't a direct quantification of how much oil is present in the sample. However, absorbance is indicated by turbidity, which is only influenced by the presence of a contaminant in the sample. So a high absorbance means high turbidity, which also means a high level of oil in the sample. And so in the LCMS, the first analysis, it was shown that acetaminophen, glyphosate, and DEET had the highest removal rates through using coconut shell biochar with iron oxide nanoparticles, removing over 60% of contaminants. The ibuprofen's most efficient removal rate was achieved through using coconut shell biochar with manganese dioxide nanoparticles. However, with the iron oxide nanoparticles as well, there were high results yielded as well. And so these pictures just show like what the data kind of look like. This is just to show like what it kind of looked like once we got it from the instrument itself and how it's converted to a bar graph so it's easily interpretable. And then that's how we got the numbers for the slide that we just saw. And then in the microscopy, which is the second analysis, the coconut shell biochar with iron oxide nanoparticles, again, had the highest removal rate of the PET microplastics, removing over 61%. And the light spectrometry, again, the coconut shell biochar with iron oxide nanoparticles, removed roughly 73% of the gasoline oil. And so overall, the most efficient method was achieved through using coconut shell biochar doped with iron oxide nanoparticles. And as stated in the beginning of this presentation, this was hypothesized. Coconut shell biochar is frequently used in soil amendment processes because it can retain nutrients and sequester pollutants in soil. These properties were expected to transfer over for water contamination, and that's why I hypothesized that it would be efficient for this as well. Iron oxide nanoparticles are also a very effective dopant, and as stated, they have a favorable surface to volume ratio, meaning that there's more area available to sequester the pollutants, and it's frequently demonstrated sequestration properties in water contamination before. For example, in my research developing the ferrosponge, the iron oxide nanoparticles were utilized to remove microplastics in oil, which led to the expectation that they would be efficient in this application as well. So with the abundance of biomass waste materials that are available for biochar, this method is very cost effective and it's easily implementable on both small and large scales. It can be used in a variety of different ways so that we can be protected from the harmful effects of these emerging contaminants in almost every scenario. Biochar is an extremely flexible and versatile material, so it can act in a variety of different ways. One example is that it could be just a simple tap water filter in our house, or another example is that it can even line large water dams and protect us from the harmful effects in those mass settings as well. Biochar could be used as a filter on its own, or it could supplement a pre-existing filter. Basically, the possibilities of using biochar are essentially endless because there are so many possibilities because of its flexibility. However, the path that I took it down was to design a sample prototype for a water filter for a household. For example, this would be used with the tap water, where it would just be placed on top or below, and the water would flow through it and it would be filtered so that we could use it. So the top compartment would have sand. This is an established method of removing larger contaminants. And since this product is aimed for everyone across the world, but specifically for underprivileged communities that don't have access to treated water, the sand would be able to remove the larger contaminants, such as plastics and metals and glass. And then the lower compartment would have the biochar, which would remove emerging contaminants. And specifically, this project showed that the coconut shell biochar with iron oxide nanoparticles is highly efficient for removing these emerging contaminants. A 0.25 micron size filter mesh would ideally be at the bottom of the filter so that the clean water can just flow out after filtration so it can be used. The prototype is still being further refined and developed. However, this technical sketch just shows some of the dimensions, the measurements, the orientations, and basically the locations of each of the sections. It's not shown in this technical sketch. However, I'd like both of the compartments to be separate. That way it can be used as needed. If the top compartment with the sand was not needed, it could be easily detached and just not be part of the product.
2 minutes.
So I'd like to further develop my prototype for my future research. On the right, you can see it 3D printed. However, again, like I said, I'd like the compartments to be separate, but I'd like to further develop the prototype by looking into different orientations, different concentrations, maybe even different filtration techniques other than centrifugal or filtration by gravity. So I'd also like to achieve a 100% removal rate, which could be achieved through testing higher amounts of biochar or testing different flow rates to see how that affected results. It's expected that this would be efficient for removing heavy metals and other contaminants, but I'd like to conduct research on that. And I'd like to investigate the reusability and durability of this further. In all of my tests, I made sure to test that this was reusable and it wasn't just a one-time usage and then you throw it out. But I'd like to investigate the durability even further to know how long this product would really last. I'm also in contact with some companies for implementing my project. I've been contacting Biosora, which is a company using biochar for soil amendment processes in sub-Saharan African countries, and I'd like to work with them so that we can implement my application of biochar to help protect those communities as well. And then I'm also in contact with KX Technologies, which is a leader in water filtration membrane development, and so I'd like to work with them for their helpful expertise, their equipment, and their materials so I could further develop my own idea of a water filter system. And so to briefly summarize, many people lack access to clean drinking water, especially in underprivileged communities. By using coconut shell biochar enhanced with iron oxide nanoparticles, we would be using an efficient method that can help protect us from the harmful effects of emerging contaminants in our water. This is a cost-effective, environmentally friendly, sustainable, easily implementable, and reusable solution, which is why it should be implemented to help keep us safe. Thank you for listening, and if you have any questions, please feel free to ask. I'll stop.